Good day everyone. For today we're going to talk about gametogenesis. Gametogenesis is a crucial biological process that ensures the continuation of life through sexual reproduction. It involves the formation of gametes, the sperm in males, and eggs in females, through specialized cell division known as meiosis. This process is essential for genetic variation, which contributes to the adaptation and survival of species. In this lecture, we will explore the two main types of gametogenesis, spermatogenesis and oogenesis, examining their stages, differences, and significance. Mitosis is primarily involved in asexual reproduction and general cell growth and repair, while meiosis is essential for sexual reproduction, producing gametes, the sperm and egg cells, with half the number of chromosomes. Mitosis produces two identical daughter cells, while meiosis produces four unique daughter cells, each with half the number of chromosomes as the parent cell. In mitosis, daughter cells inherit the same number of chromosomes as the parent cell, while in meiosis, daughter cells inherit half the number of chromosomes as the parent cell. Mitosis involves one cell division, resulting in two genetically identical daughter cells, while meiosis involves two cell divisions, ultimately producing four genetically unique haploid daughter cells. Mitosis occurs in somatic body cells for growth, repair, and asexual reproduction, while meiosis occurs in germ cells, or what we know as reproductive cells, to produce gametes, the sperm and egg cells for sexual reproduction. Mitosis produces two identical daughter cells, while meiosis generates four genetically unique cells with half the number of chromosomes, leading to increased genetic variation through processes like crossing over and independent assortment. The learning objectives for our lesson for today are the following. First, I can describe the structure of the human male and female reproductive systems and their functions. I can describe the location and stages of gametogenesis in males and females, and I can explain how hormones regulate the male and female reproductive systems. Mitosis and meiosis differ significantly in their phases as shown in the diagram, with meiosis involving two rounds of division, meiosis 1 and meiosis 2, and the formation of tetrads or paired homologous chromosomes in prophase 1, while mitosis has one round and doesn't form tetrads. Now, this is something to think about. Why do women menstruate? And why is it important? Women menstruate as part of their monthly reproductive cycle, where the lining of the uterus, or what we call endometrium, is shed if pregnancy does not occur, and it's important for reproductive health and overall well-being. Menstruation is a key indicator of reproductive health and fertility. Now, if you have irregular periods or the absence of periods, or what we call amenorrhea, it can be a sign of underlying health issues, so it's important to seek medical advice if you have some concerns. Next, we're going to talk about the organs of sexual reproduction, particularly the male and female gonads. Let's talk about the male gonads first. The male gonads or testes produce sperm in highly coiled tubes called seminiferous tubules. The epididymis is a tightly coiled tube located behind each testicle in the male reproductive system, serving as a site for sperm maturation, storage, and transport. Now let's talk about spermatogenesis. Spermatogenesis, the formation and development of sperm, is continuous and prolific in adult human males. Cell division and maturation occur throughout the seminiferous tubules coiled within the two testes, providing hundreds of millions of sperm each day. For a single sperm, the process takes about 7 weeks from start to finish. Only in spermatogenesis do all four products of meiosis develop into mature gametes. Spermatogenesis occurs throughout adolescence and adulthood. Now let us have a closer look. The spermatogonia, which is a diploid cell, will undergo mitosis to produce the primary spermatocyte. This primary spermatocyte will further undergo meiosis 1 to become secondary spermatocytes, which are haploid. The primary spermatocytes will move away from the outer wall. It will undergo meiosis 1 to produce secondary spermatocytes. During meiosis 1, the homologous chromosomes separate, resulting in two haploid or containing one set of chromosomes secondary spermatocyte. Each secondary spermatocyte then undergoes the second meiotic division or meiosis 2. Meiosis 2 results in the separation of sister chromatids, forming two haploid spermatids from each secondary spermatocyte. The spermatids then undergo spermiogenesis. It is a process of differentiation and transformation to become mature spermatozoa or sperm cells. The Sertoli cells will support and nourish the sperm cells. Now, let us talk about the parts of spermatozoa. We have the head. Okay, the head contains nucleus and acrosome or the enzymes for egg penetration. There's also middle piece which is packed with mitochondria for energy and the tail that propels the sperm forward. It allows the sperm to go swim to the egg. Let us now proceed to the female reproductive organ. These are the parts of the female reproductive organ. First is ovary. Ovary produces the egg and female sex hormones. Uterine tubes, or also known as fallopian tubes, is the site of fertilization or passageway of sperm and egg. Uterus houses the developing embryo and fetus, and vaginas, the organ of sexual intercourse and the birth canal, and last is the cervix that is found between uterus and vagina that serves as a passageway. Now let's talk about oogenesis. If there's spermatogenesis that creates the sperm, we also have oogenesis that creates the eggs. Oogenesis, or the development of mature oocytes or eggs, is a prolonged process in the human female. Immature eggs in the ovary of the female embryo but do not complete their development until years and often decades later. In oogenesis, cytokinesis during meiosis is unequal, with almost all the cytoplasm segregated to a single daughter cell. This large cell is destined to become the egg. The other products of meiosis, smaller cells known as polar bodies, will degenerate. Oogenesis begins in female embryo with the production of oogonia from primordial germ cells. The oogonia divide by mitosis to form cells that begin meiosis but stop the process at prophase 1 before birth. These developmentally arrested cells, which are primary oocytes, each reside within a small follicle, a cavity lined with protective cells. At birth, the ovaries together contain about 1 to 2 million primary oocytes, of which about 500 fully mature between puberty and menopause. Now, let us have a closer look of what oogenesis is. Oogenesis starts prenatally with primordial cells or primordial germ cells or PGCs migrating to the ovaries and dividing mitotically to produce oogonia. Oogonia then undergoes further mitosis and differentiation to become diploid primary oocytes. Primary oocytes enter meiosis but are arrested at prophase 1, a stage that lasts until puberty. Beginning at puberty, follicle-stimulating hormone or FSH periodically stimulates a small number of follicles to resume growth and development. Typically, only one follicle fully matures each month, with each primary oocyte complete meiosis 1. The second meiotic division begins but stops at metaphase. Thus, arrested in meiosis 2, the secondary oocyte is released at ovulation when its follicle breaks open. The follicle cells will burst, releasing the haploid secondary oocyte in a process called ovulation. The ruptured follicle left behind after ovulation develops into what we call the corpus luteum. The corpus luteum secretes estradiol as well as progesterone, a hormone that helps maintain the uterine lining during pregnancy. If the egg is not fertilized, the corpus luteum degenerates and a new follicle matures during the next cycle. Only if a sperm penetrates the oocyte does meiosis II resume. If fertilized, meiosis II is completed during a haploid ovum, producing a haploid ovum or egg and a second polar body. Each of the two meiotic divisions involve unequal cytokinesis, with the smaller cells becoming polar bodies that eventually degenerate. This unequal division of cytoplasm ensures that the egg has enough resources for the early stages of development if fertilization occurs. As a result, the functional product of complete oogenesis is a single mature egg containing a sperm head. The polar bodies are much smaller and often degenerate, ensuring that the egg receives most of the cytoplasm and organelles. Now, the main differences between oogenesis and spermatogenesis are first, of course, is the number of gametes produced. Spermatogenesis results in 4 functional sperm cells from 1 spermatogonium. Oogenesis produces only 1 viable egg and 3 polar bodies that degrade. The timing and the duration also differs. Spermatogenesis is a continuous process that begins at puberty and continues throughout life, while oogenesis starts before birth, pauses at various stages, and completes only upon fertilization. The resting phases also differs. In spermatogenesis, it does not have long pauses. It proceeds continuously once initiated. In oogenesis, it has a prolonged resting phases. For example, primary oocytes are rested in prophase 1 until puberty. Now let us talk about how hormones influence reproductive functions. Mammalian reproduction is governed by the coordinated actions of hormones from the hypothalamus, anterior pituitary, and gonads. Endocrine control of reproduction begins with the hypothalamus that secretes gonadotropin releasing hormones or GnRH. This hormone directs the anterior pituitary to secrete the gonadotropin follicle stimulating hormones or the FSH and luteinizing hormones or the LH. Both are tropic hormones, meaning they can regulate the activity of endocrine cells or glands. They're called gonadotropins because they act on male and female gonads. FSH and LH supports gametogenesis or the production of sperms and eggs in part by stimulating sex hormone production by the gonads. The gonads produce and secrete 3 major types of steroid sex hormones. First one is the androgens, principally testosterone, and the estrogen that is principally estradiol and progesterone. Now, let us have a recap of the differences between spermatogenesis and oogenesis. These are the main differences in gametogenesis or spermatogenesis and oogenesis. First one is the number of cells produced as mentioned earlier, the size of the cells produced, and the timing of the process. First one, number of cells produced. In spermatogenesis, the cells divide equally to produce 4 functional gametes, while in oogenesis, the cells divide unequally. Remember, there's an equal cytokinesis, resulting to 1 functional gamete plus 2 to 3 polar bodies that will later on degenerate. Next, for the size of cells produced in spermatogenesis, the sperm cells are all of equal size with equal amounts of cytoplasm. In oogenesis, only 1 egg cell retains all of the cytoplasm and the rest are polar bodies that again will degenerate. In the timing of the process, in spermatogenesis, the production of gametes is a continuous process that begins at puberty and continues until death. But in oogenesis, the production of gametes is a staggered and finite process. Now, this is the summary of oogenesis and spermatogenesis. Yeah, if you have any questions, please do not hesitate to message us. Thank you very much, everyone.